I'm Ben Redelings. I do evolutionary genetics, and my background is in biomathematics. I'm going to talk today about alignment, rate variation, and heterotachy. So first of all, let's talk about evolutionary trees. So why should we care about evolutionary trees? So this is one reason why you might care. This is an evolutionary tree that I made from some DNA sequences from HIV from humans and the simian immunodeficiency virus. So the green line corresponds to a sequence from Acolobus monkeys. The black lines correspond to DNA sequences from chimpanzees, and the red sequences are from humans. And one of the things you can see from this is that SIV jumped to humans from chimpanzees, and when it got into humans, we call it HIV. But we can see that it actually jumped 3 different times. The top time, it created a group of HIV sequences we call group O, and the middle one is group N, and the bottom one is group M, which originally stood for main. And then this has 6 subtypes: A, B, C, D, E, F, etc. So this is one way where looking at the phylogenetic trees that relate viral sequences can tell us how they have jumped species and how similar the different types are. The second thing I'm going to talk about today is multiple sequence alignments. So multiple sequence alignments are ways of taking raw data of DNA sequences from different viruses or different species and arranging them in a matrix so that we know which residues correspond. A lot of bioinformatics analyses take as inputs not raw sequence data. Instead, they take multiple sequence alignments. So my timing here, sorry. Then they take multiple sequence alignments. So we need these multiple sequence alignments to create things like evolutionary trees relating species. We can infer ancestral sequences from proteins in different species. We can try to estimate divergence times. How long has it been since 2 different species shared a common ancestor? And then we can try to estimate properties of the individual residues. So for example, the first column in this matrix has 2 residues. So it's less likely that a mutation in this DNA site would cause a birth defect compared to one of the sites that has only 1 residue. Okay, so now let's talk about the biological process that creates our sequences, the DNA sequences. So this is an evolutionary tree. I've labeled the nodes on the tree, or vertices, with black circles. The vertices at the bottom represent existing species, rat, mouse, chimp, and human, and we call these leaf nodes or tip nodes. The species, the nodes that are higher up represent hypothetical ancestors, and we call those nodes internal nodes. Each represents a speciation event by which one species splits into 2 daughter species. And then the highest node is an internal node called the root node, which represents the common ancestor population that is ancestral to all of these modern species. These evolutionary trees are essential for 2 different reasons. One of which is that all modern classification into groups, species, genus, family, order, etc., take as groups subtrees of the evolutionary tree. Each of these subtrees, all the species in one subtree share changes that occurred along the branch leading to the ancestor. So for humans, that would be things like being warm-blooded, giving birth to live young, having body hair, suckling their young. All of these would represent evolutionary changes that occurred on one branch and then are inherited by all the modern species that are children of that branch. So then for sequence evolution, we start with a sequence at the ancestor, here indicated here, ATTGCG. And then evolution proceeds on each branch in a random walk that's independent than other branches. So one type of change is the substitution or single letter change. So here this red X represents an A to C change on the ancestor of chimp and human. Another type of change can change the length of the sequences. So for here, example, we see this green X representing the deletion of 2 A's in the human ancestor And this blue X representing, the green X represents the insertion of two A's in the human ancestor. And the blue X represents the deletion of a T in the rat ancestor. Now, a multiple sequence alignment indicates the evolutionary relationships of individual letters by putting letters in the same column if they derive from the same letter in the ancestral sequence. So, for example, all of the red letters descend from a red A in the ancestor, all of the green letters descend from the green T in the ancestor, and so on. Unfortunately, we don't get to see these letters. The relationship is not directly observable. So instead, I'm going to just color these letters corresponding to their observed residue, A, C, T, or G, for the rest of this talk. Another thing that's unfortunate is that we get to observe the raw data. The aligned data is not observed, but estimated. And this raises the question, how do we estimate these alignments? So the alignments that we estimate are commonly fed into further stages of the analysis, and therefore errors in the alignment can propagate through this analysis, leading to errors in the final result. So I'm going to introduce a new or an alternative approach, which is that instead of estimating first the alignment and then the tree in a sequence, we're going to start with the raw data. We're going to estimate the alignments and the phylogeny together as a pair in one logical step. Now, this has a few benefits. First of all, because the evolutionary tree is always available, instead of just placing gaps in a matrix to indicate where a letter is missing, we can actually place insertion and deletion events on specific branches of the tree. This means that instead of alignments that look like this, we get alignments that look like this. You can see that in the green box here, we have two insertions from different sequence in different lineages. The traditional method would tend to say that because these are in the same place, that the letters must share a common ancestor, but in fact they don't. And we can see this by looking at the tree. Secondly, by doing Bayesian inference, we get to account for alignment uncertainty. There could be different alignments, not just this one shown above, and Bayesian analysis will take that into account. A joint score function says that we have this problem where in order to know the alignment, we need to know the tree, but in order to know the tree, we need to know the alignment. A joint score function solves this circularity by estimating a pair of sequences instead of trying to estimate one from the other. And then lastly, in the Bayesian framework, we can use Markov chain Monte Carlo to search the parameter space. And unlike other algorithms that have a hard time exploring this space, the MCMC is able to find both optimal and non-optimal alignments. Okay, so that's set up. So what this talk is actually about is an issue of under-parameterization in evolutionary models. So in around 2004, people were trying to estimate evolutionary trees from DNA sequences, and they noticed that one of the things that you need to include in your model is called a cross-site rate variation. What that means is that one column might evolve pretty fast and have many different letters, whereas another column that's next to it might evolve pretty slowly and have only one or a few letters. If you don't include this rate variation in your model, you can end up with a bias towards some trees that are not correct. In addition, you can get overconfidence in your results. So you could be wrong and confident, which is a bad combination. So we would like to include enough parameters so that we don't get this bias and overconfidence. Secondly, with large data sets, so when we started estimating evolutionary trees, the main limiting factor was, do we have enough DNA sequence to get a sufficient confidence? Now we have, in some sense, too much data. Because our models are imperfect, we have little biases. And when we get lots and lots of data, these little biases all go in the same direction, so that the confidence in one branch of the tree tends to be that if we support it at all, we support it 100%. So this is also a result of under-parameterization. And then lastly, we care about the biology. We don't just want to get the right answer, we want to get it for the right reason. And the way that we do that is to have a model, a statistical process that reflects the biological process going on behind the scenes. When we infer a tree or an alignment, we're inferring an evolutionary scenario that should be a plausible explanation for the data. Now, up to this point, people have generally considered the effect of under-parameterization only on the tree. They treat the alignment as something fixed and known because it wasn't possible to estimate the alignment in a high-quality statistical way. But now that we can estimate the alignment and the tree together, we can ask how it's affected by under-parameterization. So I did I'm in the process of doing a simulation study. When we have things like trees and alignments that basically say what happened in the past, we can't observe them directly. So if we want to see if our methods are getting the right answer, the only way we know what the truth is if we simulate it. So here we simulate DNA sequences evolving on a tree. The tree comes from a birth-death process, has about 500 tips, and we're going to simulate from the general time reversible model on A, T, G, and C, plus gamma distributed rates for the columns with 4 bins. And then we're using the software Indelible that adds insertions and deletions with the geometric length. Now we should be varying a whole bunch of other stuff, but I haven't. I'm in the process of doing this. So the result of this study is that when we use different software and different models, each represented by a different data point, they have different degrees of accuracy. So what we'd like to find is that for all the pairs of letters that should be in the same column, we'd like to find that all the pairs that are in the true alignment are found in our predicted alignment, and also all the ones that we predict are accurate. So the x-axis will indicates the fraction of stuff that we found, and then the y-axis represents the fraction of found stuff that's true, so accuracy. And what we see here is that the MAFT software in its default configuration has it finds about 50% of the homologous pairs, and about 50% of the ones that are found are accurate. In its more accurate configuration, the error goes to 35%, and then Prank gets down to about 25%. Bolify, the software that I've written, can get down to about 15% when it ignores variation in evolutionary rates across columns. But if we take into account a cross-site rate variation, we can make the error go down by half again to about 9%. So that seems good. However, there's one issue that I've found, and that is that in real data, we have this thing called heterotachy. Hetero means different, and tachy is speed. You might recall tachyons from Star Trek are particles that have a high speed. They go faster than the speed of light. Now, this is an alignment where I've just showing the colors, but not the individual letters, A, T, G, or C. There's a color per letter, and I'm not showing what the sequences are, but they're widely varying across the tree of life. And what we find is if we take into account rate variation, we get a different alignment. It goes from 185 columns up to 201 columns. So why would we see such a large variation in, where's this? Why would we see such a large Why would we see such a large variation? Well, what's happening is that as evolution proceeds, in some subtrees, the rate speeds up. Now we have a single column with 2 rates, and our model of a cross-site rate variation says that each column can only have 1 rate. So we take a column and we split it into 2, and now our assumptions are met again, which is that each column has only 1 rate. So how can we solve this? We solve this via a model called the Tuffley-Steele model, where instead of having only 4 states in our Markov chain, A, C, T, and A, C, G, and T, we switched to having 8 different states. Each state is split into an on and an off version, and the off version can't change to a new letter. It can only change back to the on version. Another way of representing this is that we have for every column, we have an on-off state, and then for each on and off state, we have a separate rate matrix, here indicated by Q. So Q0 for the off state, you can't change between the different letters, but for Q on, you can. So now we need to combine this with rate variation. And the way we do this is that we have a probability distribution that's a mixture of 4 different models with 25% of sites unknown in advance following each model. The rate of change between letters when you're on occurs at different speeds. So in the rightmost model, it occurs at a high speed. In the leftmost model, it occurs at a low speed. However, the rate of change between on and off occurs at the same speed. When we try to apply this model, we find that as before, using the general time reversible model with the same rate in every column, we estimate 185 columns. And interestingly, the total sum of all the branch lengths on the tree is 10, meaning that on average, there should be about 10 changes per column. If we switch to the rate variation model, we get 15 extra columns because of these column splittings, and the tree length grows to 14. This is because we're allowing more changes to occur per column in the high rate columns. And now if we try our new heterotachy model, we go back down to about 186 columns, and the tree length goes up even further. Now, one question is, does this alignment look exactly the same as the alignment that we get when we assume no rate variation at all? And if we compare the alignments, we can see that they're still a bit different, but that the alignment length is similar. What effect does this have on the tree? If you look at the tree on the left, you can see that the branches aren't very long. The tree in the middle, the branches have got substantially longer as the result of allowing rate variation. And then the tree on the right, the branches get even a bit longer again. So in conclusion, across-site rate variation models can make alignments a lot longer. These models might still create better trees because by splitting columns, they're in effect forcing the data not to violate the model. Covariant type models can shrink the alignment length back again to what we expect. I'd like to explore in the future more complex covariant models. These types of heterotachy models are still perhaps in their infancy. I'd like to thank the NSF for support.